I've told you so far only one reason why the maintenance approach seems so promising, namely that it seems to target the weak link between being alive and being dead. But there's another one, which is this. If we look at actually what damage is in concrete terms, in molecular and cellular terms, it seems that it's a lot simpler. It's not simple, but it's a lot simpler than either metabolism or pathology. Here is my first outrageous claim of the day. I claim that the things that qualify as damage, all the phenomena that qualify as damage by the definition I'm using today, can be classified into one or other of these 7 broad categories. And so if we go through these categories, we can see that they really are concrete things. Junk inside cells, what I mean here is simply molecular byproducts of normal metabolic processes that, for whatever reason, accumulate because the body does not have mechanisms for either destroying them or excreting them. And I'll come back at some length to some of them later on. Junk outside cells, same deal, but we're talking about, for example, amyloid in Alzheimer's disease. Loss of cells, too few cells, we're talking, for example, in Parkinson's disease, in the substantia nigra, cells being cells dying and not being automatically replaced by the division and differentiation of other cells. You get the idea. You can go down the list and these are real concrete phenomena. Now, of course, the outrageous statement is that this is an exhaustive list, that really everything can be fitted into this thing, this classification. And I want to give you 2 reasons for at least entertaining that possibility. The first reason is that the definition of aging involves this phenomenon of accumulation. Damage, by the definition I'm using today, has to accumulate. Now, if a protein is synthesized in a cell and it undergoes oxidation, for example, and then it's broken down, and the undamaged amino acids are reused and the damaged ones are excreted, then that damage is gone. So that's not damage by the definition I'm using today. It's not accumulating. So that means that what we can actually say is that damage can only occur in long-lived structures. So then we can say, well, okay, why don't we just look at what the body's made of? We're made of, cells and stuff between cells. What are cells made of? DNA and so on. And so we can basically just go through the body and we get pretty much this list if we do that. The other reason, which is circumstantial but still quite powerful, is it's been the same list for more than a quarter of a century. We've been looking at these things for a very long time in the biology of aging. People have been studying the roles that these phenomena might play as mechanisms dominating the accumulation of increasing pathology in old age in mammals and in other species, of course. And, we really ought to have found some more in that time, because let's face it, we've come a long way since the early 1980s in terms of our ability to analyze biological systems and figure out what they're doing and what they're made of and so on. So this is quite a strong argument too, I think. But here's the really good news, and this is my second outrageous claim of the day. I think we know how to fix all these things, in principle. Here's the same list on the left in a slightly different order, and on the right, I'm showing you why this particular list on the left is useful. Of course, there are thousands of things that go wrong in aging that qualify as damage in the way that I've described, and there are an awful lot of different ways to classify 1,000 things into 7 categories, but this particular categorization is useful because within each category, more or less the same type of treatment should be amenable to implementation against each example. So in the case of cell loss, for example, it doesn't matter whether it's in the substantia nigra or the heart or the retina, we ought to be able to use cell therapy of one sort or another to treat it, to actually reverse that damage and repopulate the cellularity of the tissue. Now, of course, different cell therapies for different tissues are different in detail, but they have a lot in common, which is why we can call this an actual field. Similarly, for eliminating the garbage that accumulates in the spaces between cells, amyloid, for example, in various different tissues, different amyloids have different molecular composition, but the same general approach that is now in phase 3 clinical trials for getting rid of it using vaccination is very likely indeed to work pretty much as well in each of these tissues as it seems to be in Alzheimer's disease. And you can go down the list and you get the same sort of logic going on. Now, that's all very well, but you probably want more detail, really, and as you can see, there's a lot of detail to be had. So I'm just going to drill down to just one of these 7 categories now in order to show you that detail does exist. I'm going to talk about this one down here because, first of all, it's glamorous, second of all, it's my idea, and also because it's really, really important for some of the most important diseases of aging. So I talked a moment ago about extracellular junk. This reason why vaccination is a cool and apparently very promising approach to addressing it is because vaccination causes phagocytosis. It causes microglia in the brain, for example, to actually engulf amyloid and get it inside the cell, into the vascular apparatus, and therefore exposed to the acidic environment with loads and loads of proteases in the lysosome. But unfortunately, there are things that already accumulate in the lysosome naturally, and the reason they do is because they're even harder to break down than what amyloid is. So what are we going to do about those things? Well, first of all, let's remind ourselves of the problem. I'm going to give you a couple of examples. Here is an EM section through an axon of a dystrophic cell, a dystrophic neuron, in an Alzheimer's brain. And you can see there's a lot of stuff here that's pretty ugly. Essentially, we have a failure of degradation here. These multilamellar structures are particularly diagnostic. Apparently, well, a lot of people think that these are caused by serial autophagy, where autophagosome double membrane structures are engulfing things that need to be broken down, and then they're supposed to be fusing with the lysosomal apparatus, and that fusion process is not happening because the lysosome has been poisoned, basically. And so the autophagosome eventually degenerates itself and gets autophagocytosed in turn a second time, and so you get this sort of thing. And then you can see it's not very happy at all, and we'd really like to fix it. This is the analogous situation in the artery. This is what used to be a perfectly self-respecting macrophage, which went into the artery wall in the attempt to process lipoprotein particles that had been stuck there, and which probably succeeded in doing so for quite a long time, but which eventually became poisoned by the contaminants, typically oxidized cholesterol derivatives, that are present at low levels in lipoprotein particles, and thereby became what's called a foam cell, this sort of undead macrophage that's the first histologically identifiable step in the development of an atherosclerotic plaque. As we all know, of course, cardiovascular disease is the #1 killer in the Western world, and probably going to remain so unless we think of something pretty clever, or unless we continue to make such pitiful success against cancer. So the question then is, what are we going to do about this? And the idea I had about, let me think now, 11 years ago, was a bit radical really, because it came from very left field indeed. Not even from anything to do with medicine at all, but from a completely different area of biology, namely environmental decontamination. So, probably 50 odd years ago now, a professor at Cambridge called Ed Gale came up with an idea that he called the microbial infallibility hypothesis. And basically, is what you've got, is what it is. If you think about a given environment, then there's going to be things in the environment that microbes can eat, and some of the microbes are going to be able to grow faster on that particular substrate, those particular nutrients, than others. So you're going to expect that in not very long at all, you'll end up with 1 or 2 species of bacteria taking over, and that's all you're going to see. You're going to see a very boring distribution of species of the bacterial ecology in this environment. Turns out that's not what you see at all. You always see an absolutely rich diversity of bacterial species. And Gale wanted to know why that was, and he realized that one possible explanation was that there are some organic, energy-rich materials in typical environments that are hard to break down, okay? That are actually somehow resistant to being broken down by most bacterial species. And if you're a species, it's one of the rare species that can break this substance down, then you've got your ecological niche, even if you're growing more slowly than your competitors that can't break it down and have to live off some other substrate. So he realized that that might be an explanation, and he thought of a good technological application for this possibility. He said to himself, well, okay, suppose we're looking at, let's say, a disused airfield that's got TNT in the soil. And you want to get rid of that TNT because you want to build a housing estate, but you don't know how. So maybe you can just go and look at the microbial ecology in that environment, and you'll find bacteria that are happily chomping away on the TNT. And then you can get them out, and you can expand them up in the laboratory and put them back in the environment in larger numbers, and your TNT is going to go away. The only reason it was there at all was because there were not enough of those bacteria in the environment naturally. Okay, that was the idea, and it turns out to work. It turns out that it's not just TNT, plastics, dioxins, rubber. You can go to the side of the highway and you will always find bacteria that are chomping away on rubber, because of course there's pulverized rubber coming off tires all the time. It turns out this is not just a theoretical concept. It really works. It is not just an academic discipline. Bioremediation, which is what he called it, is a thriving commercial enterprise. And that's pretty interesting. So what's the medical application? Well, the medical application that I realized is this. There are environments that are enriched in human remains. And the thing is, the bits of human remains that they're enriched in are not the energy-rich ones. They're enriched in like bones and teeth, right? Which of course, if you're a bacterium, it's not going to do you much good to eat those, right? They do not accumulate these energy-rich things which happen to be rather bad for our health in old age. So That could be rather interesting. It could be that that's because they've got microbes in graveyards